This video will explore Chapter 7, Cellular Respiration. Remember, Chapter 6 and Chapter 7 lecture notes will be covered on Exam 3. Use the study guide on Blackboard to prepare for Exam 3. By the end of this video, and subsequent review of Chapter 7, you should be able to discuss the learning objectives, using information from Chapter 7. First of all, the term, respiration, is defined as a series of redox reactions in a membrane that generates ATP. There are two types of respiration that occur in nature. Cells can engage in aerobic respiration or anaerobic respiration. The major difference is the requirement of oxygen for cellular growth and ATP generation. Aerobic respiration requires oxygen, and anaerobic respiration does not require oxygen. In general, aerobic respiratory processes produce more ATP than anaerobic respiratory processes. The next two slides that follow provide excellent comparisons of aerobic and anaerobic respiration. This slide attempts to show the relationship between photosynthesis and cellular respiration. Photosynthesis is a process in plants that produces carbohydrates, such as glucose. Cellular respiration is a process in non-plant cells that uses glucose to produce ATP. This slide illustrates how photosynthesis and cellular respiration are related. Essentially, both anaerobic and aerobic respiration involves oxidation reactions and reduction reactions. Oxidation occurs when a molecule loses electrons. Reduction occurs when a molecule gains electrons. Cells produce energy through a series of oxidation-reduction reactions. Oxidation-reduction reactions always occur at the same time. Stated differently, during biochemical reactions, as one molecule is being oxidized, or loses electrons, another molecule is being reduced, or gains electrons. For simplicity, we refer to oxidation-reduction reactions that occur at the same time, as redox reactions. Take the time to review this sample exam 3 question. If you have any questions about this slide, contact Dr. Flowers via email. This slide provides an illustration of a redox reaction in nature. It is clear that methane has become oxidized to produce carbon dioxide, and molecular oxygen has become reduced to produce the water molecule. The production of water is observed at the end of the electron transport chain and associated with energy production. Energy production is associated with the movement of electrons in eukaryotic and prokaryotic membranes. Electrons are passed through a series of electron carriers. Energy associated with the flow of electrons through membrane electron carriers are used to generate ATP. Later, in this video, we will describe specific electron carriers and specific cellular process, such as chemiosmosis designed to convert electron flow energy to cellular energy. An overview of cellular respiration is shown here in picture form, and using the equation, highlighted with a red box. Glucose, oxygen, and ADP are reactants, while water, carbon dioxide, and ATP are products in cellular respiration. Many electron carriers are coenzymes. Coenzymes are non-protein substances that are capable of donating and accepting electrons during anaerobic and aerobic respiratory pathways. A major electron carrier in living systems is nicotinamide adenine dinucleotide, or NAD. NAD serves as an electron carrier during glycolysis, citric acid cycle, and during electron transport. Dehydrogenases are essential enzymes that aid in the transfer electrons to NAD, converting NAD to its reduced form NAD. This slide illustrates the components of both the oxidized form of NAD and the reduced form of NADH. This slide explores the three major steps of cellular respiration. On exam 3, be prepared to list, discuss, or describe the three main steps of cellular respiration. The first stage of cellular respiration is glycolysis. Glycolysis is a 10-step process that converts glucose to pyruvate. Glycolysis only results in 2 net ATP molecules. The second stage of cellular respiration is pyruvate oxidation. After pyruvate is produced in human cells, it is broken down, or oxidized, in the mitochondrion before entering the citric acid cycle. The first part of pyruvate oxidation is called the intermediate step. The second part of pyruvate oxidation is called the citric acid cycle. Pyruvate oxidation produces 2 molecules of ATP for each glucose molecule broken down via glycolysis. The third stage of cellular respiration is oxidative phosphorylation. The term oxidative phosphorylation refers to the synthesis of ATP coupled to electron transport. The third phase involves both the electron transport chain and chemiosmosis. The third phase produces over 30 ATP molecules in human cells. This slide provides an excellent illustration of the 3 stages of cellular respiration. This figure also does a good job of reminding you about the locations where the 3 stages take place in a eukaryotic cell. There are 2 primary routes by which ATP is generated in human cells: oxidative phosphorylation and substrate-level phosphorylation. The term oxidative phosphorylation has already been defined and refers to ATP production as a result of electron transport chain and chemiosmosis. Substrate-level phosphorylation is another way ATP can be produced in eukaryotic cells. Substrate-level phosphorylation, which is shown here in this image, takes place in glycolysis and the citric acid cycle. Substrate-level phosphorylation is the formation of ATP occurring when an enzyme transfers a phosphate group from an organic molecule, for example, one of the intermediates in glycolysis, or the citric acid cycle, to ADP. Substrate-level phosphorylation events produce much less ATP molecules when compared to oxidative phosphorylation. Understanding the location of where cellular respiration takes place is important. Here, we are reminded that in humans, glycolysis takes place in the cytoplasm, the intermediate step takes place in the cytoplasm, the citric acid cycle, or Krebs cycle takes place in the mitochondrial matrix, and oxidative phosphorylation takes place in the inner mitochondrial membrane. Understand the locations for exam 3. This slide shows several views of a mitochondrion. Here, you can clearly see the location of the inner and outer mitochondrial membranes as well as the intermembrane space and the mitochondrial matrix. Remember, the citric acid cycle takes place in the mitochondrial matrix, and the electron transport chain takes place in the inner mitochondrial membrane. Also, from the figure, you can clearly see the folding of the inner mitochondrial membrane. The folds are called cristae. Briefly, cristae are folds of the inner mitochondrial membrane, and are designed to increase the surface area of the mitochondrion, thereby providing more room for oxidative phosphorylation, and hence more ATP to be produced. Glycolysis is the first stage of cellular respiration. Glycolysis is a 10-step enzyme-catalyzed process, responsible for the breakdown and utilization of the majority of the sugar we consume in our diet. If we break down the term glycolysis, we can better understand the overall process. The term glyco means sugar, the sugar is glucose, and the term lysis means destruction, or breakdown. During glycolysis, glucose, a 6-carbon molecule, is broken down to produce 2,3-carbon pyruvate molecules. This is coupled with the release of energy in the form of 2 ATP molecules. As you can see from the diagram in the right, glycolysis takes place in the cytoplasm for both prokaryotes and humans. This slide also shows the chemical reactants and products of glycolysis to enhance your understanding of the process. An important thing to remember is that glycolysis produces a net gain of 2 ATP molecules. This slide shows the 10 individual reactions in glycolysis, and also shows the chemical structure of the intermediates in the glycolytic pathway. This slide provides another view of glycolysis. From this visualization, it is easy to see that glycolysis takes a 6-carbon molecule, glucose, and converts it into 2, 3-carbon molecules of pyruvate. The green balls represent carbon molecules. In the preparatory step, or stages 1 through 5, you can see that the cell must use 2 ATP molecules. You can also see that in the energy-conserving step, the cell makes 4 ATP, via substrate-level phosphorylation. Since the cells make 4 ATP, but initially uses 2 ATP, eukaryotic cells produce a net 2 ATP molecules as a result of glycolysis. Take the time to review this sample exam 3 question. If you have any questions about this slide, contact Dr. Flowers via email. The intermediate step, which takes place after glycolysis, and just before the citric acid cycle, is a very important step during cellular respiration, and marks the initial stage of pyruvate oxidation. From this image, you can clearly see that the intermediate step involves a redox reaction, in which pyruvate is being oxidized, and NAD is being reduced. The intermediate step also involves a decarboxylation. Thus, in the intermediate step, pyruvate is converted to a 2-carbon molecule, called an acetyl group. The acetyl group binds to coenzyme A, forming acetyl-CoA. Acetyl-CoA is then transferred to the citric acid cycle for further processing. Again, after pyruvate is produced, as a result of glycolysis, it is broken down further in the intermediate step. The intermediate step occurs after glycolysis, and before the citric acid cycle. The product of the intermediate step is acetyl-CoA. Acetyl-CoA is sent to the citric acid cycle. After pyruvate is oxidized, acetyl-CoA is produced, and is transferred to the citric acid cycle. When acetyl-CoA, a 2-carbon molecule, reaches the citric acid cycle, it interacts with oxaloacetate, a 4-carbon sugar, forming citrate. The main function of the citric acid cycle is to continue the oxidation of carbon compounds, to produce reduced electron carriers, and a small amount of ATP. In eukaryotes, the citric acid cycle takes place in the mitochondrion. There are several synonyms for the citric acid cycle. The citric acid cycle can be referred to as the Krebs cycle, TCA cycle, or tricarboxylic acid cycle. As you can see from the image, during the citric acid cycle, carbon dioxide is released. In humans, this is the same carbon dioxide that is exhaled from the lungs during the breathing process. This slide shows the reactions of the citric acid cycle. From this image, you can clearly see two decarboxylations, reduced molecules of NADH and FADH2, and a molecule of ATP produced using ATP. The 8-step process of the citric acid cycle only produces a small amount of ATP via a substrate-level phosphorylation, but the reduced coenzymes, NADH and FADH2, will carry their electrons to the electron transport chain, where a large amount of ATP will be produced via oxidative phosphorylation in the inner mitochondrial membrane. Use the current slide to understand the reactants and products connected to the 8 steps of the citric acid cycle. The reactants are all shown in the blue box, and the products of the citric acid cycle are shown in the green box. Remember, since 2 pyruvate molecules are produced, you must double all of the numbers in the green box, so at the end of the citric acid cycle, you have 4 carbon dioxide molecules, 6 NADH molecules, 2 FADH2 molecules, 2 ATP molecules, 6 protons, and 2 acetyl-CoA molecules. Oxidative phosphorylation is the third and final step of cellular respiration. Coenzyme, electron carrier molecules NAD and FAD transfer electrons to the electron transport chain. This step results in a large increase in ATP production and consists of 2 parts, electron transport chain and chemiosmosis. Electron transport chain is a sequence of electron carrier molecules, or membrane proteins that shuttle electrons during the redox reactions that release energy, used to make ATP. Chemiosmosis is the production of ATP using the energy of hydrogen ion gradients across membranes to phosphorylate ADP. The electron transport chain, coupled to chemiosmosis, powers most of the ATP synthesis in cells. During glycolysis and the citric acid cycle, cells created the reduced coenzymes NADH and FADH2. This molecule enters the inner mitochondrial membrane and donates, or give up electrons, to the electron transport chain. As electrons move through the electron transport chain, they release energy. This energy is strong enough to pump protons in the inner membrane space of the mitochondrion, creating a high proton concentration gradient in the inner membrane space. As proteins move back into the mitochondrial matrix via ATP synthase, the resulting energy is sufficient to promote ATP synthesis. The previous sentence describes the process of chemiosmosis. Don't be confused here, we call it an electron transport chain, because literally, here, electrons are being transported along a chain of both protein and non-protein electron carries. The image at the bottom of the slide offers another glimpse of the electron carriers, or complexes, forming the electron transport chain. Some electrons enter the chain at complex 1, and passes to complex 2, complex 3, and complex 4. Some electrons enter the chain at complex 2, and pass electrons to complex 3, and complex 4. This helpful image reinforces your understanding of where the electron transport chain is located in eukaryotic cells. As you can see, the electron carrier complexes and ATP syntheses are embedded in the inner mitochondrial membrane. The next 2 slides provide more details about the composition of the electron transport chain. The electron transport chain consists of protein complexes, cytochromes, and ubiquinons. Remember, as electrons are passed through the chain, protons are pumped out of the matrix into the inner membrane space. Protons diffuse back into the matrix, via ATP synthase, with the subsequent production of ATP molecules. Again, the creation of a high proton gradient, and subsequent diffusion back into the mitochondrial matrix through ATP synthase, is called chemiosmosis. When electrons reach the end of the electron transport chain, or complex 4, electrons and protons finally bind to oxygen, forming water in the process. In addition to accepting and donating electrons from carrier to carrier, ubiquinone and other protein complexes can transport protons to the inner membrane space in the mitochondrion. At some point, a very high proton gradient is achieved in the inner membrane space. The proton motive force is then used to drive ATP synthesis. The proton motive force is created by the pumping out of protons by the respiratory chain complexes in the mitochondria to translocate protons through the ATP synthase complex, leading to the formation of ATP from adenosine diphosphate and phosphate. As I said before, in a previous slide, oxidative phosphorylation involves both electron transport and chemiosmosis. Another way of thinking about it is that electron transport activates chemiosmosis, which ultimately lead to ATP phosphorylation. At the end of the electron transport chain, electrons bind to oxygen forming water. You can easily see the formation of water at the bottom of this slide. We have already defined chemiosmosis in another slide, but here, we can show the process in greater detail. Be prepared to know the 3 steps of chemiosmosis. Step 1 of this process is essentially the electron transport chain. Step 2 reminds you that as electrons are passing down the chain, protons are being pumped out of the mitochondrial matrix to establish a high concentration gradient in the inner membrane space. Step 3 states that the protons re-enter the mitochondrial matrix via ATP synthase, such that ATP is produced. The image on this slide shows you the direction protons take as they travel back into the mitochondrial matrix using ATP synthase. ATP synthase is an enzyme that catalyzes the formation of the energy storage molecule, adenosine triphosphate, ATP. Remember, ATP synthase uses a proton motive force to add phosphate to ADP to generate ATP. This is an important slide. This slide shows the total number of ATP molecules produced from each molecule of glucose that enters the pathway. The last bullet shows all of the reactants and products involved in cellular respiration. This slide provides another excellent overview of the stages of cellular respiration. Further, it reminds you of the total ATPs produced in each stage of the process, and reminds you of the cellular location of part of the process. Take the time to review this sample exam 3 question. If you have any questions about this slide, contact Dr. Flowers via email. The previous slides have basically focused on cellular respiration, which is a major biochemical mechanism for producing energy for humans. However, as we stated earlier, cellular respiration is not the only way for human cells to generate energy. Another process used by human cells, that can take place in the absence of oxygen, and is therefore an anaerobic process, is called fermentation. Fermentation is known to take place in muscle cells during periods of heavy exercise. Fermentation provides a mechanism for producing energy from glucose and other carbohydrates in which the citric acid cycle and the electron transport chain is not required. Fermentation essentially consists of glycolysis and several reduction steps. Fermentation only produces a net 2 ATP molecules. Technically, fermentation is an anaerobic process, because fermentation does not require oxygen. However, fermentation is not considered anaerobic respiration, because respiration utilizes the citric acid cycle and electron transport chain, and fermentation does not use the citric acid cycle or the electron transport chain. There are many types of fermentation reactions that exist in nature. Two major types of fermentation are shown on this slide. Fermentation reactions are named based on the organic molecule produced as an end product. The end product of lactate fermentation is lactate. The end product of alcoholic fermentation is alcohol. As stated earlier, fermentation is essentially glycolysis, followed by reduction steps. When pyruvate is reduced to lactate, we call it lactate fermentation. When pyruvate is reduced to alcohol, we call it alcohol fermentation. Lactate fermentation is used by the food industry to make beneficial food products. Alcohol fermentation is used to create bread and alcoholic beverages. The next two slides show the differences between the major metabolic process discussed in this chapter. Strict anaerobes are organisms that do not use molecular oxygen and are actually killed by its presence. These organisms lack the enzymes necessary to utilize oxygen, and thus oxygen is viewed as a poison. Both bacterial cells and human cells can be facultative anaerobes. Facultative organisms have the ability to grow in the presence or absence of oxygen. When oxygen is not available in the environment, these organisms produce energy via fermentation. When oxygen is available, these organisms produce energy using cellular respiration. While some cells in the human body are facultative anaerobes, the majority of human cells are strict aerobes. That is, they require oxygen in order to live. Based on the previous information presented in Chapter 7, you now understand that oxygen is specifically required as a final electron acceptor during aerobic respiration. Most of this chapter focuses on the breakdown of carbohydrates, namely, glucose, to produce cellular energy in the form of ATP. This slide focuses on the fact that your cells can also use other macromolecules, such as proteins, lipids, to produce energy. This slide also informs us that the products of catabolic reactions can also be used in anabolic or biosynthetic reactants to make essential cellular components. Overall, cellular respiration plays a role in catabolic reactions, designed to break down macromolecules, but also provides the basic carbon precursors for anabolic or biosynthetic reactions. During catabolic reactions, ATP is used to make essential cellular molecules for growth. As stated previously, human cells possess distinct and specific metabolic pathways for the breakdown or oxidation of all the four major macromolecules: nucleic acids, proteins, carbohydrates, and lipids. The image on the right shows you that oxidation products of carbohydrates, fats, and proteins are transferred to various stages of the cellular respiration pathway, namely glycolysis and the citric acid cycle, in order to generate ATP. At this point, you should understand that ATP is very, very, very important in order for living organisms to survive and thrive on Earth. This image displays the major pathways that oxidize, or break down carbohydrates, fats, and proteins. Sometimes glucose is needed in the cell, but energy is not. In this case, gluconeogenesis is utilized. Gluconeogenesis is the process of synthesizing glucose from non-carbohydrate precursors. Gluconeogenesis takes place in the cytoplasm. It is a way creating new glucose from the products of its breakdown. Its main function is to supply blood glucose in case of carbohydrate deficiency. Every molecule in the human body must be regulated to maintain homeostasis. Feedback inhibition is used to ensure that glucose levels and ATP levels are sufficient. This slide explains a basic feedback regulation mechanism for both glycolysis and the citric acid cycle. In both instances, ATP and a metabolic intermediate binds to an enzyme in the pathway. Typically, this enzyme is required in the first, second, or third steps of the biochemical process. For example, ATP binds to phosphofructokinase in glycolysis, and ATP binds to citrate synthase in the citric acid cycle. Take the time to review this sample exam 3 question. If you have any questions about this slide, contact Dr. Flowers via email.